Hello, my name is Dr. Roderick L. Roll. Today I will talk to you about carbohydrate metabolism. We are going to break down sugars. One type of sugar that you are familiar with is fructose. Fructose can be catabolized into an intermediate that will directly be linked to glycolysis. If you look at the image below, we have fructose, which is a sugar. It will be converted into fructose 1-phosphate. Then fructose 1-phosphate will be broken down into glyceraldehyde and dihydroxyacetone phosphate. Dihydroxyacetone phosphate is step 4 of glycolysis. Then that will be converted into glyceraldehyde 3-phosphate, which is the reactant for step 6 of glycolysis. Another sugar that will be catabolized is glucose. So glucose catabolism is a cumulative function of 4 metabolic stages called cellular respiration. These 4 stages include glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. We will discuss all 4. The first one is glycolysis. Glycolysis occurs in the cytoplasm of a eukaryotic cell. Glycolysis is nothing more than the conversion of glucose into pyruvate. This requires 10 enzymes, and it takes 10 steps to do this. You will have to know all 10 steps of glycolysis. This picture illustrates that. This picture illustrates the rest of glycolysis. The first thing that occurs is glucose is going to be converted into glucose 6-phosphate. In this image, we have a 6-circle molecule. Glucose has 6 carbon, so each circle represents a carbon. To the left of that 6-circle molecule, we have ATP. We have a pentagon-shaped structure with 3 little black circles. Each circle represents a phosphate group. So step 1 requires energy and requires a glucose molecule. This next slide shows the product of step 1. This completes the product of step 1. So glucose is converted into glucose 6-phosphate. ATP comes in, donates a phosphate group to carbon 6, and ATP turns into ADP. The next step, glucose 6-phosphate, is converted into fructose 6-phosphate. The next step is going to require an additional ATP. This concludes step 3. ATP gives a phosphate group to carbon 1. Therefore, changing fructose 6-phosphate into fructose 1,6-bisphosphate. So far, we have used 2 ATP molecules. Step 4, we will split fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So let's look at the molecule to the right, and we're going to take that through the rest of glycolysis. Then we will come back and obtain the molecule to the left of the fork. That is dihydroxyacetone phosphate. We will convert that into the molecule that's on the right. Let's do that. Step 5 is converting dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. The next step, we're going to need a inorganic phosphate group. This phosphate group is not attached to a carbon. We will also need ADP to be an acceptor, and it is going to accept electrons from glyceraldehyde 3-phosphate. So the sugar that has the 3 circles is glyceraldehyde 3-phosphate, and it has a phosphate group located on carbon 3. We're going to add another phosphate group to carbon 1. Step 6 converts glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. Notice that the phosphate group, the inorganic phosphate, was added to carbon 1, and we had a redox reaction occur. NAD was converted into NADH. In order for step 7 to occur, we are going to require ADP. It is going to receive a phosphate group from the previous sugar that was produced in step 6. This is referred to as substrate level phosphorylation. So 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. That is step 7. ATP is created in step 7. Step 8, 3-phosphoglycerate is converted into 2-phosphoglycerate. All we did in this step is move a phosphate group from carbon 3 to carbon 2. Step 9, we will convert 2-phosphoglycerate into phosphoenol pyruvate. In step 10, we're going to take phosphoenol pyruvate and convert it into pyruvate. This step will also create ATP. Do not forget, these steps occur twice. Steps 6, 7, 8, 9, and 10. Listed here are the actual names of the sugars. Glucose is converted into glucose 6-phosphate by hexokinase. Glucose 6-phosphate is converted into fructose 6-phosphate. Fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is converted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Step 5, dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. Step 6, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. Step 7, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. Step 8, 3-phosphoglycerate is converted into 2-phosphoglycerate. Step 9, 2-phosphoglycerate is converted into phosphoenol pyruvate. Step 10, phosphoenol pyruvate is converted into pyruvate. In column 2, labeled glycolysis, place the appropriate number in each square. It has to be a number, 0 through 100. Make sure you fill in column 2 that is labeled glycolysis. The 3 stages of cellular respiration include the synthesis of acetyl-CoA. We're going to call it the prep step. Krebs cycle. It can also be called the TCA cycle or the citric acid cycle. And finally, we're going to look at the redox reactions. All of this occurs inside of the mitochondria. Glycolysis occurs in the cytoplasm. The prep step is nothing more than taking pyruvate and converting it into acetyl-CoA. Now, in order to do this, we're going to release one of those carbons. Pyruvate contains 3 carbons. We're going to release one of those carbons, and the product that's going to be left over is going to be a 2-carbon molecule called acetyl-CoA. But before we create that, we're going to have to add a coenzyme A molecule, and we have to go through a redox reaction. So remember, when we do a redox reaction, the sugar is going to give electrons to NAD. So therefore, when we take 1 pyruvate and convert it into 1 acetyl-CoA, we have 1 decarboxylation, and we're going to produce 1 NADH. This is a drawing of that reaction. This step is accompanied by a decarboxylation, redox, and the addition of a coenzyme molecule. Coenzyme A is derived from vitamin B. In column 3, labeled prep, place the appropriate number in each square. The Krebs cycle is part of cellular respiration. It is where most of the energy comes from during reduction of coenzymes such as NAD and FAD. This occurs in the cytoplasm of prokaryotes, and it occurs in the matrix of the mitochondria. This is the Krebs cycle, or the citric acid cycle, or the TCA cycle. You will need to know all of the substrates and products of this cycle. So let's draw it like we drew glycolysis. This is going to be a cycle because we're going to generate the starting product with each cycle. So we're going to take a 4-carbon molecule called oxaloacetate. We're going to unite it with the 2-carbon molecule called acetyl-CoA, and we're going to create citrate. That's step 1. Citrate is converted into isocitrate, step 2. NAD is going to come and receive electrons from isocitrate. NADH is made, decarboxylation occurs, and we're going to create a 5-carbon molecule called alpha-ketoglutarate. In step 4, alpha-ketoglutarate is going to give electrons to NAD. So NAD is needed, and coenzyme A is needed. Alpha-ketoglutarate gives the electron to NAD, therefore reducing it to NADH. Coenzyme A is going to be added, and we're going to have another decarboxylation occur in step 4 of the Kreb cycle. the product that is produced is acetyl-CoA. A inorganic phosphate is going to be used in step 5. It is going to convert ADP into ATP. So succinyl-CoA becomes succinate in step 5. FAD is required for step 6. Succinate becomes fumarate in step 6. There's another redox reaction that will occur, and we will produce FADHH. Fumarate will be converted into malate in step 7. NAD is required for step 8. For step 8, malate will be converted into oxaloacetate. This is another redox reaction, and NADH will be formed. We will create our starting product each time this cycle occurs. This is a list of all steps. Step 1, step 2, 3, 4, 5, 6, 7, 8. Step 8, you will have malate will create oxaloacetate with the use of an enzyme. Write all of these substrates and products. In column 4, labeled Kreb, place the appropriate number in each square. This should be the answer. Now let's look at the structure of the mitochondria. The mitochondria is composed of 2 membranes. It has an outer membrane and an inner membrane. The inner membrane is going to be folded, and these folds are called cristae. The cristae will increase the surface area. In between the inner membrane and the outer membrane is the inner membrane space. Inside of the inner membrane is the matrix. So the mitochondrial membrane is going to move electrons, therefore creating this high hydrogen concentration in what is called the IMS, the inner membrane space. Inside of the inner membrane is several complexes. These complexes will create the electron transport chain. We have 4 multi-protein complexes, and each one will carry electrons. Electrons will be donated from NADH or FADHH. They will be donated to the inner membrane of the mitochondria. Electrons will move down the multiple protein complexes, and eventually the electrons will be handed off to oxygen. If oxygen is not present, this process will stop. Let's look at one of these multi-protein complexes. It is called complex 1. Complex 1 contains flavoprotein and a flavin mononucleotide called FMN. This is derived from vitamin B2. It also contains an iron sulfur protein. This is only one of the complexes. NADH is going to give the electrons to FMN of complex 1, whereas FADHH will give its electron to FAD of complex 2. So NADH becomes oxidized by releasing the electrons to FMN. FMN therefore becomes reduced. FMN will become oxidized when it gives the electron to FES. Then FES will become reduced. Then FES will become oxidized when it gives the electrons to Q. And this will continue to move the electrons down this electron transport chain. Eventually, the electrons will end up on the oxygen molecule, therefore creating water. So FMN becomes oxidized by releasing the electrons to FES. FES is therefore reduced. Most of the remaining electron carriers between ubiquinone and oxygen are proteins called cytochromes. Their prosthetic groups contain iron. So if somehow you're able to remove the iron from these cytochrome complexes, the cytochrome complex would no longer work. Therefore, you would not be able to move the electrons down the electron transport chain. Let's look at another way to stop electrons from moving down the electron transport chain. Cyanide is a poison that can be obtained from crushed apple seeds. Cyanide can restrict the movement of electrons down the electron transport chain. This will hinder the production of the hydrogen gradient. Remember, as electrons move down the electron transport chain, hydrogen will be pumped into the intermembrane space. Cells use energy released in redox reactions to form proton motive force. This is also called chemiosmosis. So in this image, we are looking at the product of redox reactions. NAD will donate electrons. The electrons will move from complex to complex that is embedded in the inner membrane. Hydrogen will be pumped into the inner membrane space. This creates proton motive force. This is also referred to as chemiosmosis. A total of 34 molecules of ATP are formed from the catabolism of one molecule of glucose during chemiosmosis. So chemiosmosis is once we have created this high hydrogen concentration, that will produce 34 ATP molecules. The two coenzymes, NADH and FADHH, will both produce energy. This energy is based on the amount of hydrogen that is pumped into the inner membrane space for each of these coenzymes. NADH will make 3 ATP because it will pump 10 hydrogen into the inner membrane space. FADHH will not produce as much hydrogen into the inner membrane space. Therefore, the energy production is lower. Only 2 ATP will be produced. NADH versus FADH, oxidized versus reduced image, is listed here. Notice how in the NADH reduced version, you have added that extra hydrogen. In the FADHH version, you have added 2 extra hydrogen. This slide shows you that the high hydrogen concentration, chemiosmosis, is going to spin the turbine. The spinning of this turbine is going to generate ATP. This is created through redox reactions. Now let's look at fermentation. When oxygen is not present, fermentation will occur. The purpose of fermentation is to produce NAD. NAD is used in step 6 of glycolysis. If NAD is no longer present, glycolysis will stop. In column 2 and 3, place the appropriate number in each square. So fermentation is going to generate waste for the cell. The products that are produced, such as ethanol, are not useful to the cell. That is waste. The purpose of fermentation is to generate NAD. If NAD is not present, the cell will not be able to produce ATP. Remember, energy is produced in steps 7 and 10 of glycolysis. If you cannot proceed past step 6 because NAD is not present, you will not be able to generate energy. Listed in this slide are several waste products for cells that go through fermentation. Propionibacterium will produce Swiss cheese. Aspergillus streptococcus will produce lactic acid, which is used to make soy sauce and cheddar cheese and yogurts. Yeast, Saccharomyces cerevisiae, is going to be used to make ethanol. The ethanol is a waste to that cell. Clostridium is used to make isopropanol and acetone, and Escherichia will create vinegar.